Hello, I am Dr. Aizaz from MedicoVisual.com, and in today's visual lecture, we will discuss the development of skin. So, after the embryonic folding, we know that the outermost covering, this pinkish one in this 3D model, the outermost covering is the ectoderm. Now, the ectoderm is basically the derivative of the epidermis of the skin. Basically, the skin has two main parts, the epidermis as well as dermis. So, outer layer, the epidermis, it is derived mainly from the ectoderm. So, let's take a part of the ectoderm. Let's just focus on this part and let's enlarge this part of ectoderm here. And here, let's see that what is happening. So you see, initially the ectoderm is single layered structure. A single layer of cuboidal cells are there. These are basically the cuboidal cells, cuboidal epithelium, which is making up this ectoderm. And of course, um, inner to these cells is the basement membrane. So they are attached with their basement membrane. So basically, this is the simple cuboidal epithelium. Now, what will happen? First of all, these cells, they will proliferate to form yet another layer outside. They will form yet another layer. So, what are these two layers now? Right now we have two layers. So, outside, not outside, inside the inner one, the deeper layer. This is basically the layer of simple cuboidal to low columnar. Simple cuboidal to low columnar cells. So simple cuboidal epithelium, and this epithelium actually it will not transform, rather I should say it will proliferate and it will form all other layers of epidermis of the skin. So it is the generative layer, and it is the basal layer. It is basal layer. It will form all other layers of the skin. So it is basal layer. So along with that, of course, outer to it, we have this temporary layer. It will later be shed off, and this is called periderm. It is called periderm. Now, what is the function of periderm? Um, Actually, the embryo, it is floating right now. The embryo is floating in the amniotic fluid. So, one of the function of these peridermal cells is that they are involved in exchange of material. Right now, there are no blood vessels here. There is no dermis here, and there is no blood vessel. There are no blood vessels in these developing epidermal cells. Even in the mature epidermis, there are no blood vessels. But later, there will be this dermis will develop, and within the dermis, blood vessels will develop. So right now, these developing epidermal cells, they cannot get nutrition from here because there is no dermis, and from the embryo they cannot directly get the nutrition, the oxygen, some chemicals, some electrolytes, some important chemicals that they require. They cannot get from the embryo because the dermis is not there, and dermis will bring the blood vessels. So, in short, there is no coverage of blood vessels here. So, embryo is all around surrounded by amniotic fluid, and amniotic fluid contains some important nutritious structures as well. It contains some chemicals, some electrolytes, right? So, actually, what will happen initially, very early stage, not at the later stage, of course. So at the early stages, the periderm, it helps in exchange of material. Peridermal cells, they help in exchange of material between the amniotic fluid and the developing epidermal cells. So that's important thing. That is one function. Another function may be, some authors believe that there may be some protective function of the periderm as well. Later on, I will tell you that they will be shed off and they will form the cheesy cheesy material outside the skin, vernix caseosa, and that has the actual protective function. But still, even before the vernix caseosa forms, even at that stage, some authors believe that periderm directly may have some protective function. They protect developing epidermal cells from the harmful substances present within the amniotic fluid. So right now two layers are there. Then what will happen that these basal cells, this germinative layer, it will proliferate again and form yet another layer between these two. So now we have these three layers. So now we have three layers, and we have basal layer, we have periderm layer, and in between these two, this middle layer is actually called intermediate layer. Very logical name and very easy to understand. So this is intermediate layer. Here we have these basal cells, right? Just I have shown two basal cells and two peridermal cells. Now what will happen that these basal cells proliferate, they will divide, right? They will divide, and the newly divided cell, the daughter cells, they will move upward like this, and they will form a new layer outer to these basal cells. And these cells, they will continue to proliferate like this until 3 to 4 or sometimes more layers are formed above it. So let's say here, for the sake of demonstration, three layers are formed. So now basically all these cells, they basically arise from the basal cell layer. This basal cell layer is the master creator, right? It is the master creator of all other. It is the parent of all other layers of the epidermis of the skin. Now here another important thing. Okay, before that, let me tell you what happens with these cells. Now these cells, they will establish connection with their neighbors, right? They will form strong connections with their neighbors, and these connections are called desmosomes. Along with that, they will form special proteins, protein fibers called uh, tono tonofilaments, right? And these are basically the keratin filaments. So keratin filaments or tonofilaments develop here. I have shown in this illustration this greenish yellow fibers. These are tonofilaments, and these are basically the keratin filaments. They have several different functions. I will not go into detail in this lecture. That's a complete different topic of histology that we will not discuss in this course of embryology, right? So now, before going further, let me tell you something about these basal cells. So typically what happens that if this is a cell, uh, it may divide into two cells, and these two cells will go on, they will become specialized, and they will become very different from their parents. They will become very different from their parents, right? They will become specialized, and they will become a different variety of cell. But here, in this case, in case of proliferation of basal cell, something slightly unusual occur. I said slightly because this phenomena is present in other tissues as well, and that's very important phenomena. Let me explain this thing to you. It's very important. So here, let's suppose here this is the basal cell, right? Now this single basal cell, it basically proliferated and it divided into two cells, right? So it divided into two cells like this. So what actually happened that one of the cell, it remained there. It remained here in this layer, in the basal layer, and the other cell, it moved forward, it moved superiorly, or it moved outward, it become more superficial, it formed a superficial layer. As you can see, it came here, right? So this cell, it formed the other type of cell. I will tell you what type of cell is this. It formed the other type of cell, as you see here. But uh, the one of the cell, it remains there. So basically what this basal cell is doing is that it is maintaining its own population. So if this basal cell proliferated in such a way that it formed two daughter cells, and these two daughter cells went on to form two different types of cells, then ultimately the population of basal cell here, that would have been lost. And if that population had been lost, that would be a disastrous situation, because now we cannot get more cells. No, we cannot get more skin cells, so no new skins will be able to develop. So these basal cells, they will remain even in adult life. So if there is some damage to skin, these cells can proliferate and they can form the epidermis of the skin. Even without the skin damage, there is normal skin turnover. Normally, the epidermis is shed and new epidermis is continuously being formed by the beauty of presence of these basal cells. So these basal cells are basically forming a stem-like pattern. So rather than one, they are forming their own variety, and then they are forming a new variety. Then their own type of variety and new type of variety, right? So that's how they are acting as a stem. So these are called stem cells, and these stem cells are present in many different tissues. Almost every tissue of our body has the stem cell variety of some kind, which provides these generative and proliferative cell. These cells have regenerative as well as proliferative capability. Well, again, I didn't want to go into details of this, but somehow I think you need to understand these important concepts. So here, actually, what happened that these desmosomes, they interconnected these cells. Now, these desmosomes, typically, they should not be very prominent, but actually, when you create the sections, when you make the sections in the histology lab and you observe it under the slide, then what will happen that some cytoplasm is lost. So as the cytoplasm is lost, the cell shrinks, these cells, they shrink, and these desmosomes, they become prominent and they form a spiny appearance of the cell. Basically it's an artifact, but nonetheless it creates this spiny appearance of the cells. So that is why this is the most prominent feature of this layer, these type of cells, that is the presence of these spiny processes, which are basically nothing more than histological artifact due to the presence of desmosomes basically. So that is why these due to the presence of these spiny cells, this layer, the prominent feature of these cells is presence of these spiny appearance. So that is why this layer is called, this group of layers basically, it is called stratum spinosum. And by the way, the basal layer is called stratum basale, and stratum simply means layer. So stratum basale and stratum spinosum. Well, actually, I said that it is called stratum spinosum, but what if I tell you this is technically wrong? Why I am saying this? This is technically wrong because right now all layers of the epidermis of skin are not established, so it is not appropriate to right now label it as stratum spongiosum, not spongiosum, stratum spinosum. Right now it should be called intermediate layer, as in the 3D model you had seen, that it should be called right now the intermediate layer or stratum intermedium, right? So it is basically right now intermediate layer, but later on, of course, as the all the layers are established, here at this place the stratum spinosum will be there, which have this characteristic feature, right? So these characteristic features are such that, let's suppose if you pinch the balloon with these clips, cloth clips. That's how the stratum spinosum, the cells of stratum spinosum form. Actually, these clips are basically like desmosomes. So here these cells are tightly adherent. So these are tightly adherent at the desmosomes. So that is why these cells are, as let's suppose you draw a bit of air from this balloon, you deflate the balloon. So that's how at these places where it is these the balloon is pinched with these clips, so here they will form the sort of spiny processes. So that's how the stratum spinosum develop, you know. So let's move forward. Now, what will happen? Now, you may be tempted to think that the cells of stratum spinosum, now they will proliferate to form another layer outside it. And even I know many people that think that cells of stratum spinosum, they proliferate to form the outer layer, which is basically stratum granulosum. But this is not correct. Actually, still the stratum basale will proliferate. What is the reason for this? The reason is very simple, and I have discussed this time and again, but briefly, let me tell you that as cells become more and more specialized, most of its DNA is turned off, so it is very difficult for that cell to divide. So, here in this less specialized stratum basal cells, the DNA is mostly uncoiled, so it is very easy for these cells to divide. So these cells can divide, but here the cells are becoming more and more specialized, so it is difficult for those cells to divide. So very easily the cells of stratum basal, they divide again like this to form yet another layer. Not yet another layer, basically more than one layer, 3 to 4 layers are established like this, and these are now stratum spinosum. So do we have more than one stratum spinosum? Well, no. Actually, here now we have stratum spinosum. So original stratum spinosum. That is why I said that we should not label it stratum spinosum right now, because it has been now replaced by yet another stratum spinosum. So that's how these cells are deceiving, my dear medical students. So don't get deceived by them. Learn and have a clear concept with MedicoVisual. So enough of this self promotion and marketing. Let's get back to the lecture. So here basically these older cells, these are younger cells, these are older cells. So these older cells, they have become more wise. So as they have become more wise, actually they have become more specialized. That's how we should say, they have become more specialized. And as they become more specialized, along with this characteristic presence of desmosomes and the spiny appearance, they have yet another important feature that develop. Basically, they will form certain protein granules inside them. You see these whitish specks here in this in this diagram, you see. So what are these? These are basically the aggregates of some proteins, some important proteins, right? These are not membrane bound, these are basically aggregates of special proteins that are translated by these cells. One of the important protein is filaggrin protein. Filaggrin, I hope I am writing the spelling correct. Filaggrin protein. So one of the function of filaggrin protein is that they aggregate the keratin molecules, right? So let's suppose if here is the keratin, not keratin molecule, keratin filaments rather I should say. So keratin, these are the keratin. So if a filaggrin comes here, what it will do is that it will sort of, you know, make make these keratin filament come close together. So they will become close together like this. So keratin filament will aggregate together. They will come near to each other, and they will become more intimate, right? And they will become aggregate. And initially these were filaments, these were tonofilaments. So these tonofilaments, they aggregate to form tonofibrils. These aggregate keratin filaments or tonofilaments. These are called tonofibrils. Aggregate keratin filaments, aggregated keratin filaments or tonofilaments are basically called tonofibrils, and that is the function of filaggrin proteins. So filaggrin proteins, they cause compaction, they cause sort of compaction of these proteins, and as it happens, the cells, they also shrink, and they shrink and become more of squamous shaped, they become more of squamous shaped, right? So now at this stage, these cells, they also have these spiny processes, desmosomes. But along with that, the characteristic feature which is not present in other layer, the characteristic feature which is not present in other layers of the epidermis is the presence of these granules. Desmosomes are still there, but they have another unique feature which is not present in other layers. That is the presence of these granules. So that is why it is called stratum granulosum. But still I would not label it stratum granulosum, because they will move forward and they will form yet another layer. So then we will have another stratum granulosum basically, right? But I am just giving you the idea. We do not have only these granules, but some lipid granules also develop like this, and then they come out of these cells, they are secreted out of the cell, and they will form lipid covering outside the cell, which will increase the water permeability, not increase, decrease the water permeability. Basically, it will form the water barrier, it will form the water barrier of the skin, right? So basically these yellowish one particles that I've shown here, these are called lamillar bodies. They are basically certain lipids that reinforce the plasma membrane in such a way. Outside the plasma membrane, they form another lipid layer which forms the permeability barrier, water barrier of these cells. So that's how it works. So now let's move forward. So these basal cells, they will proliferate again. Don't think that these cells, they will proliferate again. Our heroes of the show, the basal cells, again they will proliferate to form a few more layers, right? And now what will happen, that these oldmost cells, the most old cells, these outermost and most old cells, what they will do is that they will lose their organelles, and they will even start to lose their nuclei, and they will become more flattened to form an outermost layer like this. So you see that they have lost their nuclei, and they have also lost their organelles, which are not shown in this illustration. This is basically a special type of cell death or apoptosis, in which the cell, not the complete cell is lost, the cell membrane is still intact, but only the nucleus is lost, and the organelles are lost. Now, these are basically just the bag of cell membrane which are outside, reinforced by the lamellar bodies, and inside that they are filled with lots of keratin, and this keratin, it is aggregated and it is compacted by these filaggrin proteins, right? So by the way, I forgot to tell you that in the stratum granulosum, these whitish protein aggregates, these are called keratohyalin keratohyalin granules. So there are not keratohyalin granules, but rather these granules, from these granules, the filaggrin protein, they came out, they separated, and they compacted the keratin filaments into keratin fibrils or tonofibrils, and that's how these sort of dead cells form. Not sort of, these are actually dead cells. The outermost layer of your skin is basically the dead cells. These are, and this process of conversion into these dead cells is called keratinization. So that is why this squamous epithelium, the stratified squamous. This is the stratified squamous epithelium, and the stratified squamous epithelium, which outside contains dead cell or keratin filled cells or keratinized cells, it is called stratified squamous. Very logical name. Stratified squamous keratinized epithelium. Now you may be thinking why it is called stratified squamous, because we also have the cuboidal cells in the lower layers. Actually, it's a tradition, or it's a convention in histology that whenever you name the stratified layer, you have to see the outermost layer, and outermost layer here is squamous layer. So that is why it is called stratified squamous keratinized epithelium. There is non-keratinized epithelium as well, but we will not discuss about that. The epidermis of our skin is basically stratified squamous keratinized epithelium. So let's move forward. Actually, what happens that in thick skin, some of the proteins from the keratohyalin granules that is transformed into a translucent protein, and that protein is called elidin protein. So elidin protein is transformed from the proteins of these keratohyalin granules in some layers of thick skin. So this protein is basically translucent protein. It's a clear protein, it's a translucent protein. So that is why some of these cells, they become translucent like this. So if there was some object behind them, you could see those object. You could see that object. So these are translucent cells, and these translucent cell is called stratum lucidum. It is only present in the thick skin, and some authors argue that it is basically a part of stratum corneum. By the way, the outer cells, these outer dead cell layer, it is called horny layer. Not horny in that sense, but this horny comes from the horn of animals. So horny layer, or it is also called stratum corneum. So some authors argue that this stratum lucidum, this translucent layer, it is basically a part of stratum corneum. So it's a deeper layer of stratum corneum. So that's how all the layers of the skin develop. And by the way, what about periderm? Actually, later we will learn that the sebum creating glands, they develop and they form whitish oily secretion, and within that oily secretion, these peridermal cells are shed. So outside the skin, there will be a specialized whitish oily material, and that oily material will contain these peridermal cells. Along with that, some other dead cells may be there. Then along with that, some hair cells may be, not hair cells, actually hairs will be there. Some shed hairs will be there, and some other substances may be there. And this whitish cheesy whitish material is called vernix caseosa. One of the most important function of vernix caseosa is that the embryo is floating inside the amniotic fluid, and amniotic fluid is very rich in some harmful substances. For example, bile salts, for example, urea. And even if there were no harmful substances, even the pure water, if you dip your hand or any part of your skin in pure water, ultimately the water might breach the barrier of your skin and disrupt your skin. So at this time, when especially when the skin is immature, not fully developed, so that's at that stage it is very important to protect the skin from the, especially the developing skin, from the harmful chemicals and even plain water of the amniotic fluid. So it provides the protection from amniotic fluid. Vernix caseosa, one function is that it provides the protection from the amniotic fluid. And embryo is all the time and fetus is all the time floating in the amniotic fluid. Later on, when the baby comes out and takes the bath, not baby cannot take the bath himself, rather their parents or medical staff, and they wash the baby, this vernix caseosa is removed, right? So yeah, one of the function is that it provides protection from the amniotic fluid. Another important function is that during the normal vaginal delivery, the baby can come out rather easily from the birth canal. So, right, that is an important function, that it provides a lubricating function. So, now let's write the names of these layers to review it. So, this is stratum basale. This is now stratum spongiosum. Spongiosum, not spongiosum, stratum spinosum, right? And even though we still have these spiny processes here in these cells, the desmosomes are still present in these cells. The most prominent and unique feature here is the presence of granules. So this is granulosum, stratum granulosum. Then in thick skin only we have stratum lucidum. This may be 1 to 2 layers, and outer dead layer is called stratum corneum. You know what? Our skin is covered by dead bodies. That's very unique. And by the way, this is a genius plan of nature, I think. This reminds me of a movie scene. In some movies, a character is holding a dead body and it is using that dead body as an armor against the bullets coming towards the character. You might have seen such movies, such action movies, I'm pretty sure. So that's how it happens. So here you can see a bit of longer illustration, a bigger illustration here. So actually, as these cells proliferate in the limited area, what will happen that they will form sort of folds like this, these inward folds like this. You see these inward folds. And later, dermis will form. By the way, these folds are called epidermal ridges. So later, dermis will form here, and not later, actually, all these processes, they are going on side by side. They are going on almost simultaneously, but I am explaining the development of dermis later. So dermis will form such infoldings here, and those are called dermal papilla. They will interlock with these epidermal ridges. And by the way, these ridges, these are also the basis of presence of unique fingerprint pattern in your body, in your fingers basically. So yeah, so now we have another problem. Our skin, it is exposed directly to the sun, and sun contains some harmful radiation, UV radiations, and UV radiations may damage our skin. That may even be carcinogenic, that may cause DNA damage, and that may trigger development of cancer. So actually we have natural sunscreen in our bodies, in our skin, and that sunscreen is provided by special cells. Neural crest cells, they arise here, they come here. And neural crest, don't ask me what are neural crest cells. You can watch the lecture on neural crest cells. We have discussed those numerous times. I don't want to make this lecture any longer, so let's not go into neural crest cells. So neural crest cells come here, they come to reside here, and here they will transform into some specialized kind of cells, and these cells are basically the factories that create sunscreen, not actually sunscreen, basically the dark pigment called melanin pigment, the darkening agent of your skin, and they give this pigment to surrounding cell. We will discuss briefly how they give this pigment to surrounding cell, but that's how it works. So these cells are basically called melanocytes, and the origin of melanocyte from embryonic point of view, you need to know the origin of melanocyte. So origin of melanocyte is neural crest cells. This is important. So along with that, of course, as the skin is outermost covering of our body, so that is why it is also often exposed to germs. We have this barrier of entry, the outermost keratinized layer. We have this barrier to entry, but still some germs, some other particles that may enter into skin. So we have a component of immune cells here as well. So basically these are antigen presenting cells called Langerhans cells. So Langerhans cells, these are basically like these are basically cousins of macrophages. So Langerhans cells, these are basically antigen presenting cells. I will not go into their details, but their origin is from bone marrow. They arise from bone marrow. Their origin that that is required for you to learn in this class of embryology, that is important for you. For the functions and histology, there should be separate lecture series that I will do it later in my life, hopefully, if I get the chance. So yeah, we have melanocytes, we have Langerhans cells, and then finally we have yet another type of cells, and these are called Merkel cells. These are basically the touch sensors. As the nerves develop, the nerve will have connection here. So basically we have these mechanoreceptors or touch receptors, and these are Merkel cells. Now, what is the origin of Merkel cells? Merkel cells. Actually, their origin is not clear. Some authors believe that they arise from neural crest cells. One theory is that they arise from neural crest cells. The other theory, the other hypothesis, rather, I should say, is that they arise from these epidermal cells in this area, the basal cells. So, these are basically, they may be specialized epidermal cells. So either they arise from neural crest cells or these epidermal cells, they become specialized to form the Merkel cells. So their exact origin is not clearly known. So all this was about the development of epidermis. Now what about the dermis? The dermis is a little bit more complicated, and first we will discuss what is the origin of dermis. So if we cut a section of embryo, we have this paraxial mesoderm here. This is the paraxial mesoderm, and paraxial mesoderm is split into block of tissues, and these blocks are somites. I will quickly skip through this part because I have discussed it time and again, especially in the development of musculoskeletal embryology. So I think most of you should already know this. So these somites are developing here, and then these somites, they divide into two parts, the dermomyotome and sclerotome. So this part is dermomyotome, and this part is sclerotome. Now, even the dermomyotome has these lips here, and these lips will form the muscular part. So, this is basically the myotome part, actually. So, myotome part. Now, from this myotome part, the dermatome part will separate, and this dermatome part, it will form the dermis. But will it form the dermis of all the skin? No, it will not form the dermis of all the skin. Actually, it will form the dermis of back region only. It will form the dermis of back region only, except in the chest region, where it will form the dermis of anterior part as well. But most in the other parts of the body, it will form the dermis of back part of the body. So, what about the dermis in other regions, especially in your limbs? So in the limbs and in other regions, the dermis, it develops from this lateral plate mesoderm, basically the outer layer of lateral plate mesoderm. This one, it is called somatic layer. This is somatic layer of somatic, not somatic, sorry, somatic layer of lateral plate mesoderm. It is also called somatopluric mesoderm. Somatopluric mesoderm. So this will form the dermis. So two origins of dermis, the dermatome part from somites in the back region and in the chest area, number one. And number two, the lateral plate mesoderm, basically the somatic part of lateral plate mesoderm. These two are the origin of dermis of the skin. And how the dermis develops, let's go to the other 3D model to understand it. So here, let's suppose this is lateral plate mesoderm, or it may be dermatome part, depending upon the area of the skin. So let's suppose it is the lateral plate mesoderm. If it was the back region, it would be the dermatome. Very easy to understand. So cells from the dermatome or from the lateral plate mesoderm somatic layer, they will proliferate like this. These are basically the mesenchymal cells. They will proliferate and they will transform, they will become specialized, and they will release certain chemicals such as glycosaminoglycans, hyaluronic acid, and other chemicals that will pull water here. So that will pull water here, and they will spread far apart, a bit far apart from each other like this. So this glycosaminoglycans and water and other substances here, this is basically called ground substance. It is actually the connective tissue. It's nothing unusual here. The dermis is actually the connective tissue. Within the connective tissue, these cells are there, and cells are interspersed loosely within a ground substance. Ground substance is mostly water, but along with water, these are the hyaluronic acid, glycosaminoglycans, and other proteoglycans, other chemicals that pull the water in this region. Along with that, these cells, they will become more specialized, and they will generate reticular and collagen fibers in this area like this. And that's how actually the dermis develop. Finally, one thing I forgot to make in this 3D model is the development of blood vessels here. Blood vessels will arise here, and of course, nerves will arise here, nerves will come here, and that's how the dermis develop. And by the way, the dermis here, it is sort of going inside the epidermis like this. So these are basically dermal papilla. These are dermal papilla, and these pegs of epidermis here, these are epidermal ridges. Epidermal ridges. So these dermal papilla and epidermal ridges, they interconnect with each other, interlock with each other to form this peg-like structure, interlocked structure. So that's how the skin develop. And another important thing, that here some of the fat cells, they develop. Some of the fat cells, they also develop in the deeper layer. The origin of these fat cell is likely the somatic layer of lateral plate mesoderm, but it is not widely discussed in the literature, so I'm not 100% sure about this. So that's how the skin develop. In summary, a very short summary, if you talk about that, skin has epidermis and skin has dermis. If you talk about epidermis, it arises mainly from ectoderm, and beside ectoderm, neural crest cells and bone marrow cells, neural crest cells and bone marrow cells from bone marrow, they also contribute by providing the Langerhans cells, melanocytes, and possibly the Merkel cells. But mainly it is the ectoderm that forms the epidermis, most of the epidermis, right? And if we talk about dermis, it developed from the mesoderm. Which mesoderm? It is either the dermomyotome from paraxial mesoderm, the dermomyotome from paraxial mesoderm, or somatopleuric mesoderm from lateral plate mesoderm. Lateral plate mesoderm. So this is most important crux of this lecture. If you want to forget everything, just at least remember this thing about the development of skin. This is the crux or executive summary of this lecture. Now for this lecture of embryology, it is not important that you understand the melanocytes, but I love this mechanism so much that I wanted to explain something about the melanocytes. Now here is a melanocyte, and this 3D model is not very accurate. Actually, it's not this uniform distribution of melanin pigments. Basically, the melanin pigments, they are created near the nucleus, here the ribosomes and Golgi apparatus, that will synthesize the melanin pigment, and that will create some vesicles that will contain the melanin pigment, and these vesicles will go into these processes. And now, how come these melanin pigment will go into the surrounding cells, the surrounding epidermal cells? Actually, these processes, these are literally eaten up by these cells, epidermal cells. This process is called phagocytosis. So they phagocytose these parts of these processes, and they engulf, and ultimately the melanin will reach in front of nucleus, and it will form sort of umbrella over the nucleus, thus protecting the nucleus. Isn't it beautiful mechanism of nature? I just love it. So finally, here is the 3D model of the skin that I have created for this lecture. You can observe this. Um, yeah. So 3D section of the skin here, and you can see the water barrier formed by lamellar bodies in the outer layers of the skin. And if I make it translucent like this, you can see nuclei, and you can observe that outer keratinized layer, they do not have nuclei. These are basically dead cells. So this is the dermis with these fibers and these fibroblasts here. This is melanin pigment. If we label this 3D model, if I label this 3D model for you, this is the stratum basale. This is stratum spinosum. And you can see these white particles here, white specks here. These are basically the stratum granulosum. And finally, we have stratum corneum. Let's suppose it's a thin skin, so we do not have stratum lucidum. If it was thick skin, here, somewhat here, there would have been another layer that would be stratum lucidum. So these are all the layers of the skin. And inner to it is the dermis. You can see the dermal papilla. These are the dermal papilla, and these are epidermal ridges here. So that's how the skin develop. I hope you understood, and you have developed a great concept from this lecture. Thank you so much for watching this video. Have a great day. Goodbye.